Next, we will have a automation panel discussion with a few people from PNNL. And so if our panelists just want to come up and introduce yourselves, I think maybe we can.
Gonna be online.
All right, we have Moses online with us. There we go. So if we want to just go ahead.
Sure. Good morning. My name is Erin Bredeweg. I am a biologist here at EMSL. My background is mostly in fungal genetics and biology.
All right, hi, I'm Bram. I am an earth scientist, but maybe more accurately a microbiologist here at PNNL. So my background has been in soil microbiology, but I've sort of made a recent foray into AI designed experiments.
And good morning still. My name is Micah Miller. I've been at the lab since, I guess, about 2008. And in most of that time, I've been working on what I call a practical laboratory automation. It's like a realistic thing, almost like a cruise control for your car. Like what's the pain point for the person doing the experiment, and what can we do to actually help that one thing? It's not really pushing the frontiers of science, but it's improving people's lives. I think so. I've been doing that most of the time. Technically, I'm a chemist. Here at PNNL, we work on so many different projects, so many different things. A lot of times a physicist won't be working on physics, a chemist won't be working on chemistry. So my title says chemist, but I am not a chemist.
Moses online, can you introduce yourself?
Hello, everyone. I'm Moses Obiri once again, and I'm a data scientist here at the lab in the Energy and Environment Division. My background is in statistics, and so here at the lab, I apply machine learning, statistics, experimental design sort of methods to solve biological and environmental questions.
Wonderful, thank you. For this first question, why don't we just go in that same order we introduced ourselves in? So the first question, can you each describe how you are thinking about using automation and AI ML tools with your experiments?
All right. So automation and AI ML tools are very applicable to genomics. So a lot of the interest that I have in understanding biology and how the genome works exists in linking phenotype to genotype. And automation will incredibly speed up the process of being able to screen mutants, mutant libraries, CRISPR-type libraries, to characterize broadly how an organism behaves in a whole slate of different conditions. AI/ML is already being used, so we have massive omics capabilities as we collect different data types on a single or consortia of organisms. Trying to put all of that data together becomes overwhelming, but if you have AI/ML tools that are able to take in information from different sources, structure it in a way that links different data types together, that really improves the power of what you get out, even just as a hypothesis for your next experiment, and makes biology both faster and more powerful.
So I use AI ML tools more, I think, it's more to the traditional sense. I'm not using the generative models that you guys, I think, have touched on at the later points in the week. It's more of the, I hate to say traditional ML, because that still seems new to me, but it is more in that vein. A lot of the projects that I'm doing that are using AL are also using the systems that have been put out by Paul Jensen, who gave a talk earlier this week, I think, with you guys, or was that actually earlier today? That was today. And so some of those philosophies that he spoke about and pain points for automation are the ones that I try and carry forward as well. And so what I try and use it is to understand how we can design experiments differently, because if we have an automation aspect, then it changes the way we should maybe be exploring the problem than if we have to do everything by hand. And so there are some things that are easy and some things that become difficult from that. And they're different than if you're doing everything the old-fashioned way. And so my work is trying to, like Erin, build up the best data that can be used interoperably so that we can eventually combine it with some other data stream.
Yeah, so I mostly approach automation towards are we doing anything in the lab at midnight? Are we doing anything Saturday night? Is the lab accomplishing anything? Right? Our sponsors, they pay for fantastic instrumentation. It's just sitting in the lab unless there's a pilot flying that instrument. And if we do have a pilot, an expert operator, what's going to happen when that person retires and we have to hire somebody else to fill that spot? Is that data going to be reproducible? Well, if we have an automated system, it should be, but if we're currently still locked into, individual vendors selling specific products, we can't connect, product A to B because those are different companies. We got the Android and the iPhone over here. That's a big problem for us. And so if we can just bridge those little gaps, then suddenly we can start exploiting a lot of these investments that we've made. We can start allowing people to break away from the mundane tasks that they're doing all the time. There's nothing more painful to me than when I hear somebody say, "Oh, that's a routine thing. We'll just have the new guy do it. We'll just have the new hire do it." No, those are miserable jobs. Like nobody wants to sit there for hours on end pipetting something, and you don't even come up with that accurate of a result. And so anything we can do to alleviate the labor that goes into performing lab work, which is real, right? That's a real thing. It's not just another loop in a solver. Like actually moving fluid from one sample to another. There's a real sense of value when you see something move in the laboratory. As far as AI ML, yeah. So I mostly use like classic kind of ML. Mostly for computer vision systems, being able to interpret what your surroundings are, what's the context, right? If I ask my robot to pick up a pipette, does it need to know what a Sharpie looks like? Is there a chance that those two things could ever be on the same benchtop? There is. It happens all the time.
Thank you. Moses?
Yeah, so on my part, I don't perform any experiment myself per se, because lazily I'm just a data scientist, but I focus on sort of the data collection workflow. So there are two pieces. In terms of AI ML, I focus on the data collection workflow and also sort of the model design. So in terms of the data collection workflow and sort of streamlining the data collection and ensuring that we have high quality inputs. And then in situations where there are no deterministic model to feed into sort of the automation process, coming up with also high quality surrogate model to then help the domain scientists sort of answer the question. So two pieces, the data collection part and then the surrogate modeling part. That is where I think I use AI ML.
Great, thank you for explaining that. And then while we're on a similar topic, let's move on to this question of, since we're in EMSL Summer School, it might be interesting for people to hear what kinds of skills or background they should consider developing to work in automation broadly. So considering our broad backgrounds, can we all speak to what you would suggest people work on in terms of skill sets? And maybe let's go in the opposite order, start with Moses.
All right, so for data scientists, I think the obvious one is, building a strong foundation in either computer science or engineering or some statistics or some sort of applied math degree is important. And I must also say this, based on my experiences with working with domain scientists, if you can also build a foundation in any of the domains that you want to work in. So if it is biology or material science or even biology, various sort of aspect of biology that you want to sort of work in, that will be sort of a big plus. And then there's also sort of the coding skills. So being able to code in either Python, R, SQL, or any sort of coding languages is a must these days. And then aside from the technical skills, there are also non-technical skills. So being open, being able to work with domain scientists across board, being able to sort of take feedback and then sort of work on whatever you are working on I think is a key non-technical skill that you need to be able to work in this space.
Yeah, so one thing there that I would try to echo is the communication skills. Is being able to work with whatever you're trying to automate. It's a guarantee there's somebody out there already doing it, right? And if you have some new idea, it's probably a slight refinement on something that's already being done. The buildings are full of experts, and they know more about that one specific reaction or that problem than you'll probably ever know, because we can't all be the smartest at everything. And so being able to walk into their office, hear their concerns, and then relay back a vision for how you think you could help them, or what direction they could move with their project, who the new customers might be for that project, and being able to coordinate a team on that same vision and move forward, I think is probably more important than any specific technical skill. Maybe as an entry-level employee, you have to have some kind of technical skill to get a foot in the door. But eventually, the technical things just kind of becomes the static. What you really need to focus on is the big picture. Like, do you have a vision for this? Can you follow through on that vision? Who's the team that's going to help you? Because all of these things get so big that no one person actually does all the work. So you need to be able to rally a team.
I think that's actually a really good thing that I was going to say something very similar is that in almost any institution that you might be a scientist in, probably the single biggest thing you'll be able to need to do is to come up with a new concept and obviously execute that concept. But creativity is actually one of your biggest assets. Science is actually a creative discipline, sometimes rather than a technical discipline in a lot of ways. But being that this is, you guys are thinking about that entry-level step into science, or many of you are, not everyone here, but having one language that you are very proficient in for coding, and then being able to use that as a way to translate commands into a different language with the sort of generative technology. So I think probably for all of you, though, the generative AI tool set is something that you all will have to embrace, right? Because that is how now we're reformatting many of our AI systems and autonomous systems to try and be based off of a generative model of some type, right? So that a human doesn't even have to code. They could maybe just prompt in English. But you still have to ground truth, and you have to check everything that a generative AI system gives you, right? I find logical inconsistencies all the time in the output that I get, right? And so if you run a bunch of things that the AI generates for you, you're going to get something wrong, and you have to be able to ground truth that. And so if you have some coding base, you will be able to actually get quality output. So that's probably the mix of the coding language and then the familiarity and ability to put together a generative model are probably going to be what are big skill sets for you guys.
Yeah, I would echo all of those things. Big themes are communication between scientists and experts, but also communication with your systems, whether it be AI or automated systems. There are being conversant in multiple fields, whether that's biology, and really being able to articulate what your scientific question is, or communicating that to a statistical expert so that you can design the best experiment, communicating that to a coder who can speak to the actual instruments doing the experiments, and really you are the human in the loop. And that's the thing that I always come back to, is you have to be able to ground truth. Just as Bram said, you have to be able to sanity check what's happening and make sure that it is actually answering the question that you want to ask. And certainly for biology, there are starting conditions that may dictate the value of your experiment. So at least at this stage, having some beginning status and understanding that the basic biology that you're really querying is really essential. And yes, I'm speaking for my field, but I think that the themes are consistent across many different concerns when you're trying to initiate this.
Yeah, I think we can all agree having the biology, the chemistry knowledge is important, but you don't need any physics. I'll say no physics necessary. There's no panelists up here who will defend the physics.
Great points. I guess it's also important for me to remember that I am the human in the loop. So I'm the human here. Do we have any questions here in the audience yet? Otherwise, we can move on with a few prepared ones. We don't have any in the chat yet.
I was just going to suggest that if anybody has a specific experimental question that you're really thinking about applying what you have learned in this summer school, or the things that you're planning to learn for your other research, if you have a specific question that you wanted to discuss, that would be fun to
Unless it's
hear
a
about.
physics experiment.
All right, we do have a question in the chat now. Determining gene function and mapping genotype-phenotype relationships is a big research question in plants for food and energy.
Mhm.
Is there any work you do in this area or resources you have?
I primarily work in fungi, but I definitely do work on experimental questions involving plants. So one of the big concerns in some of the research themes recently is understanding how plants interact with soil microbes, as well as endophytes. So organisms that end up affecting how a plant is expressing its genes, what those phenotypes are. There are certainly plant scientists here. I'm not one, but there is work, I think, more extensively going on at some other national labs like Oak Ridge that do more extensive plant phenotyping. It is of interest to understand how genotype affects outputs. Like that's a giant question in biology. The other purpose of implementing some of this automation and very large scale data collection is so that we can predict what organisms are going to do in specific environments. And that includes certainly at first microbes that you can grow easily in 96 well plates. But this I'm expecting to expand to many other experimental types. We also need to consider what's happening in the soil. How does that affect what is happening? That plays into what is happening on the automation front in Monet, which you probably have heard about some this week. So plant phenotypes matter. I don't have more to say. I hope that answered your question at least partially.
Yeah, that was great. Does anyone else have anything to add for that one?
I mean, I think that the genotype to phenotype is we call it one of the grand challenges in biology, right? Because if you had a perfect sequencer, the theoretical perfect sequencer, and you get every scrap of DNA from an organism without any sequencing errors, you still would not be able to predict the phenotype. There is too much other biological information that creates the emergent concept of the phenotype. And so what a lot of us are trying to do here is figure out what other pieces of data, what other omics can we combine, or do we need to combine to be able to get a sort of complete sense of what a phenotype actually is.
I'm also gonna, I wanted to also throw in metabolic modeling, because this is another part of be very important computational involvement and in phenotyping. So when we collect all of these data, we are now trying to construct a predictive understanding of what is actually happening as far as movement inside the cell. And that again is probably could be described as a chemical phenotype, but it is an aspect that is largely driven by AI/ML, and it continues to develop. And I saw some hands up before.
Yeah, I think we had a question over here first.
About the ethics. In this week, we learned that this is our responsibility. Oh, this is not working.
You have to flip it up for it to be on. Just double check. Thank
Can
you.
you hear me?
Yeah.
Okay. So in this week, we learned about the ethics. Okay. We are responsible of the output that we are going to deliver, right? So, but what about the ethics around the world using this technology? Because we just have an idea so far about how powerful the AI is, how we are in the world controlling this to don't be used to build technology that is going to damage people. So are the scientific talking about this, or what are your thoughts about that? I don't know, did you get my point?
I mean, people are already, not people in this room, surely, but people are already designing technology that harms people. And so the AI tools will just be ways that scientists on both tracks of the ethical, spectrum will be using. So certainly the emergence of generative AI doesn't change the mission objectives of the national labs, right? Just because we have AI doesn't mean that we suddenly get to do unethical things, right? We still have the same goals that we had 5 years ago, or the same positive societal goals that we had 5 years ago. So for many scientists and institutions, this shouldn't change those aspects.
And I would also add that there is always a personal responsibility, right? That especially here at PNNL, if your boss, if your employer says, we want you to invent the doomsday device, like anybody can walk away from that. You can always opt out and say, you know what, personally, my views don't align with that project, and I need to go find something else because that's not I'm not going to go poison people. Not really poison people, but just.
In addition to personal ethics, I also sit on the Institutional Biosafety Committee, and that is overseen, or what that does is oversees any research that involves recombinant DNA or recombinant proteins, and that we report directly to the NIH. So NIH has guidelines about production of toxins and oversight of any research that involves toxins, giving new characteristics to organisms that might involve increasing virulence or toxicity. There are now also movements into oversight of protein design and expression. So in anything like AlphaFold, are you designing proteins that maybe have damaging properties? That is coming into place. So we have developing oversight into how proteins are designed and whether they can actually be expressed. So there is involvement and concern about that. So it's not all happening in free space. This is a known concern, and it's something that certainly for any funded research lines that come through the NIH is being looked at.
Okay, so I'm gonna go with my question, and I'm gonna kind of put the conversation back to the phenotype genotype problem. Like you said, that it is very difficult. I'm also working on that with fungi, and there's not that much information in many species. There was one talk here today earlier saying that there is in microbiology like good information about like 10 species, and then the rest is like not that much known. So then do you use here, like some AI tools or some of machine learning that we've been learning here today, like this week, to like help maybe connecting some data or dots, like to get more information on these things that they don't really have that much information.
Well, there is an emerging initiative in the DOE space, especially, to make large multi-omic data sets interoperable, right? To combine efforts from places like EMSL and JGI, the Joint Genome Institute, to be able to put lots of differently formatted, differently stored, and data with different ontologies into something that can be put into a larger predictive framework to maybe start to have better data in biology. And it's something, if you maybe saw Paul talk earlier today, he might have also described the field of biology as actually somewhat data poor in terms of it's very hard to get data from lots of different experiments together. And you could spend your whole PhD on just one meta-analysis, for example. So I think I'm hopeful that in the next 5 or 10 years, the ability to combine many different data repositories together, we'll be able to have deeper data about model organisms that is more transferable to non-model organisms, or perhaps be able to pull in disparate sources of data for the non-model organisms to say, actually, we do have some information about this organism that you didn't really know because we're looking in the right database. And in 10 years, I hope that's an unacceptable answer, that the information is there for you ready, so you actually do have what you need to better match your genome to phenome.
Yeah. And I might also add that I think in my sort of experiences so far, I think in some of the work that I've done in this biology domain, there's been an emphasis on using Bayesian approaches. And so sort of, I think just to add to what is just said, sort of using LLMs to sort of search for information online and then combining it with your existing data. I think that I'm currently working on a project that we did exactly that. We are doing some type of Gaussian modeling, and so the data is limited. So we use LLM to search online, gather information about whether they are proteins or lipids or whatever metabolizer you want to study, and then combining it with your existing data, no matter how limited they are, to then get, I think, a better picture of what you're trying to study. So I think, yeah, going forward, LLMs, I think are powerful when it comes to this space where we know data is very hard to come by.
Great, thank you for those answers. Do we have another question? Oh, make sure your mic's on.
Yeah, so this is kind of more of generic question to like lab automation and stuff like that. So based on what we have had throughout this week, I've realized that when it comes to lab automation, the PNN is kind of spearheading the research, and like a lot of cool things are going on here. So what are the efforts of PNN? I'm talking about EMSL, PNN labs to kind of share this expertise, especially with the rural communities and some labs with less resources, expertise, and funding, to make sure that every lab can actually have this idea or get access to these resources, and be able to kind of make it like a common goal for every scientist to say, okay, this is what you're doing, this is how you can automate your lab and stuff like that.
So I would say that a lot of our hardware problems are really problem dependent, right? We make a lot of custom things just for one. It's a constant struggle as we're trying to decide how many resources do we want to apply to a general case, right? Do we really want to build out the capability of this platform so that we could use it for anything? Or because we're fund limited, do we really just target it towards this one thing? Most of the efforts we do here are completely open. If there's some particular intellectual property, there's some patent that's made, the patents are great. I got a patent on the L shape. Like, how crazy is that?
Yeah.
Anyway, so we share technology. We commercialize some of it if it happens to be something that somebody else can get paid a quarter for. But the big problems that we have is that a lot of the problems we're trying to address are large scale problems. Like, we want to handle 5,000 samples a month. And like a small lab has different issues. And so our conveyor system, our shipping and receiving systems, those aren't necessarily going to be applicable to somebody who just wants to try this out in their small university laboratory. To me, I think that's the biggest problem with transferring these techniques out into the rest of the world. We try to write up a lot of the things that we discover, but I think you'll find that when it comes to hardware and actual really automating things in the lab, it's very difficult to find journal articles, right? We talk about normal research. We go peer-reviewed research. We can look up all kinds of things. We can learn from what other mistakes other people made. When it comes to designing the robot that's going to reach under the table and turn the screw and open the vacuum, those things are very difficult to find. And I think it would be great if we had a better venue that we could actually share specific engineering designs.
I mean, there's also a bit of a competitive angle too, right? Because if we figure out a way to process 5,000 samples in a day and no other labs have that angle, then that makes us more competitive when we're trying to win funds from sponsors. And that applies to university labs too, because university PIs also have to compete for grants as well. So if they have a capability that gives them an edge, as much as you maintain an open source principles, you still want to be the person that people go to for that technique. In terms of, I think, transferable skills, then what I think the most thing is, given that some labs are large and some labs are small, some labs are not going to need a robot to automate things. Many labs might need an AI awareness so that they can maybe design their experiments in a more thoughtful way. And so that requires a sort of broad expertise and base skill level for AI tools across the scientific population. And that's probably one of the big things that we'd like to be able, that would be one of the big wins, I think, in the next decade, is if everyone understood how to use some level of AI to just to speed up their work and to take some human busyness out of the element.
I think the thing that I would add is, at least on the raw data side, and actually speaking also to the question about phenotypes and genotypes that came just before, is that the data that PNNL generates and that EMSL generates is fair. It's openly accessible. There's a huge amount of data, and it may be that using AI/ML systems with curated data sets that come from a unified source, or that come from sources that are maybe not the most popular hit on a ChatGPT research assistant, would generate hypotheses. So, and where I see this being really useful for implementation of automated systems in smaller labs. I mean, I'm sure you've heard of the molecular phenotyping center that will be opening here in the coming years, maybe M2PC. It is going to be slated for just generating very large amounts of data about new organisms. So trying to define how an organism will respond, predict how it will behave in a specific environment, is going to be open to small labs, to anyone who applies for access. There are ongoing research efforts that generate programs to probably interface both with data and with automated systems that may be available. I mean, you probably have to find a specific project. You may need to correspond with a specific scientist, but the national lab is here as a resource. And so yes, there is the competitive edge issue, but at the heart of it, I think we're all scientists and we are interested in information and research.
Hi, thank you so much for coming today. And I have a question that is kind of specific, but I would like to know if maybe you have used any, or if you know if there's any machine learning tool that can predict the function of certain genes, because there's still a lot of genomes that have hypothetical genes, and basically all the hypothetical proteins, and basically all the tools that Broca use or Rust is BLAST. So I don't know if there's a machine learning tool that can predict the function of these hypothetical proteins. So
there is a tool that has come out of PNNL recently called SNECMER. So SNEKMER, right? Like so part of the word is Kamer in there, but it's SNEC because, SNEC and it's Python and it's cute, So that is a tool to help, and I can say it again if you wanted to take that note. I'll say it at the end, but it's so like you mentioned, a lot of the gene finding tools are predicated on BLAST searches or other things that are using hierarchical Markov models or hidden Markov models, right? So HMMs. And so they're very good at finding things that are true, and they're hard at finding things where the homology doesn't match anything very close enough. And SNECMER tries to cast a broader net, and so you get many more hits on what an unknown function might be. I know that it is, I think, intended and developed for microbial genomes, but the problem is consistent for fungal, plant, animal genomes as well. Even for model organisms, there's many genes we just don't know what their function is. So that would be a tool to look at.
Oh, thank you so much. Can you repeat the name?
So it's SNEK, SNEKMER.
Yes, SNECMER's great. Some other things that I use pretty frequently are Paper Blast. I don't know if you've heard of Paper Blast, but it was developed by JGI. What it does is you can put in a gene identifier or a protein sequence, and it will blast all of the literature and come back with orthologs from other organisms that have literature support for new functions. And so it'll also return a percent identity. So like you have your gene sequence of interest, we'll say it's like 67% similar or something like, and then you have some information about it. So certainly for eukaryotic genomes, that is another useful tool. There are also more and more built-in functions in UniProt that will use AlphaFold and three-dimensional shapes of proteins to predict functions based upon structural similarity instead of just sequence similarity. So there are lots of things, and those are not unified. I think that's actually a need to have a consolidated source of information that might integrate lots of different sorts of sources to try to predict function. But I am an experimental scientist, and so I would always want to say, yeah, but can you experimentally verify that? So go back to the lab and see what you can find.
Moses, do you have anything to add to that?
No, the only one I know is I think JREM, which I think predict functions relating to certain genes using, I think, test mine information from PubMed abstract. So maybe I can put the name.
I didn't.
Yeah, let me put the name here so that let me type it to everyone. I think JREM.
Thank you, Moses.
Yeah.
And also, I'll just go ahead and add too that there is a tool called Clean Contact. Clean Contact, yeah, it's a long acronym, but that is specifically for enzyme EC number functions. And that takes in the amino acid sequence as well as the three-dimensional structure. So it does both. So that's just an example of how you don't have to purely use the structures. There are ways this project was specifically deep learning, but there are ways to incorporate more than one modality into your work like that.
Hi, my question is coming back to genotype to phenotype related. My question, I think, would be more related to how to do analysis. So I'm a plant biologist working with multi-omics data sets to see how environment affects the plant's development with epigenetic and genetics involved. So when there's epigenetics involved, there's like a lot of factors involved. And my idea for my analysis is to use deep neural network for DNA methylation, open chromatin, and gene expression to like combine the analysis in a way that which be able to predict in future that like this, but if the temperature is changing or the drought conditions are happening, these changes might be happening. Obviously, experimentation is needed to check those. So my question is, during analysis, would it be easier to put all those information in one particular model, or like making different models for each analysis, and the result is the comparison point? That makes sense. And along with that, like the factors involved, like temperature is changing, site is involved, all those factors also.
So one thing that took me a while to learn, so coming from more of an experimental background and struggling with ML methods a little bit during my PhD, and then modeling a little bit more in my first postdoc, was that if you're, it strikes me sort of a modeling or computational focus question. And if the question is, do I do this or this or this? If you're modeling, the answer is yes. You do all of it, and then you find a way to compare what gives you the best, unbiased, most accurate, precise, whatever your metric of success is, answer. Especially if it's something new, which is what research typically does. Whereas with experiments, we have to be, sometimes you have to be choosy too, right? If you have only a certain number of computational hours to use on the cluster, then you pick one or the other. But in a lot of cases, for besides the biological data, I've been able to say, oh, I need to understand this parameter, and I want to compare this tool with this tool and this other library, then the answer is yes, I run all of those tests and compare them. So separate models, a joint model.
Yeah, and I also add this, that when you are faced with this kind of question, do I do everything at the same time, or do I separate them? I always start with the separate, simplest one first. Most of these deep learning models, right, suffer when there are multi-collinearity issues. And so when you have all these things done between, at times your predictions, not at times, most times, your prediction may not be the way you want. And so what I always advise is that you start with the simplest one first. So if I'm trying to remember what exactly you wanted to do, but if there are sort of three separate questions, you start with maybe a simple model for each of them first. Look at their predictions, see that they make sense. If they make sense, then you can then think about ways of then aggregating them. And so always our advice, start with the simplest one for one research question first. Look at the prediction, try to validate the result you get from each. If they make sense, then you then think about how to combine them. And then thinking about them, you think about, okay, are they multi-collinearity issues? Should I be reducing the dimensions of some of the variables that I have, things like that? But yeah, our advice, start with the simplest one first and then build from that.
My question is about training the robots. So I know that usually we use math models or camera to learn the movement, but also I know that using like a gloves, we can train the robots with the human movement to know how much pressure or the angles and whatever. So are you working on this, continuing this path? What is the progress that you are having, or is it not working? What is the best way to train the robot for you so far?
So I think when I approach these problems, especially with robotics, I look at it as what is the current operator doing? Almost like if you set up a camera and recorded somebody in their lab, in their office all day long and played it back at high speed, like what are they doing? They're reaching out, they're touching a keyboard, they're kind of wiggling a mouse a little bit. In the office, that's all you do. And so if you can design some system, we'll call it a robot, that can wiggle that mouse and can type the keyboard, it can do the office work, the same work that a person would do. You just wheel the chair out of the way, you wheel the robot in. All night long, it's replying to your emails, it's scheduling meetings, it's doing a great job. In the laboratory, it's a little different because we have so many more things that are delicate that we need fine motions on. But again, if you can reduce the problem, right? You put the starry vision of oh, the robot's going to do so much more than what a person could do. You just reel it back to say, let me duplicate what a person could do. Like, what are the motions? And I think the most success I've had is simply by duplicating exactly, and I've done this before in the lab where you stand with one hand behind your back and you say, okay, I need to do this. Let me see and step through, run my own simulation, if you will, of exactly how I would do that and what are the directions my wrist can move. And then you can start worrying about, getting the manipulators to move how many gears, how many axes, how much travel do you really need? But it's so easy to go to the human interface, which is what all of our instruments are already designed for, right? We love to have digital interfaces and we control it over the cloud and run these things. But the toaster that you buy in your kitchen, it has a button, right? It's already got an interface. And so if you just have something that can operate the interface that's already there, then we're hardware independent, right? It doesn't matter who made that instrument. It almost doesn't matter what that instrument is meant to do because it's got that interface. We just have a robot click the mouse, it can do anything.
I had a question on the computational side. Are you aware of like any other methods aside from like Bayesian optimization, sensitivity analysis of like implementing machine learning tools or like AI tools to not replace necessarily, but enhance or model certain biological or physical phenomena as well? Like aside from the data management side?
Should I? When it comes to optimization, apart from, I think, currently what I'm a big fan of Bayesian optimization. And so I'm working on current project with Erin. That is what I'm using. So that is my go-to. But there are also genetic algorithms out there, optimization algorithms out there that are broadly used. I'm also a big fan of particle swarm optimization. It's also, it works as well as Bayesian optimization. Especially if you are dealing with sort of high dimensionality, particle swarm works very, very well. But I think in the simplest case, if your system is well designed, you have very small number of sort of variables, your equation is very deterministic. I always go with gradient descent. It's very interpretable, it's simple to do, and it works all the time. And so depending on how big your system is, I think I will start with gradient descent. It's easy to do, it's intuitive, easy to follow. And then there are a lot of, like I said, a lot of genetic optimization algorithms are there, aside from Bayesian optimization, that you can also use.
So Paul Jensen's group at who does have Bacteria AI, I think they use Gaussian process regression as well, but they also, their published paper uses a bagged neural net, which I think the bagging system helps the neural net generalize better than otherwise a neural network would do. So it does better on test data.
Hello, I have a question about training models on simulated data. So I know like we have sometimes limited data availability. So let's say if we come from basic principles and simulate some like basic physical principles and build simulate some data. And I'm wondering like what considerations I should take into account when training the model on the simulated data? And if someone had experience, like maybe like what's the percentage of simulated plus, real data I have to have or something like that? Thank you.
We have to make it messy. That's something easy to make really clean data that it suits all your criteria, but most data, especially from biology, probably chemistry too, has got a lot of mess to it. So you want to be able to handle weird stuff.
Yeah, and I think there are two things going here, right? So there is what you said, the data simulation part, where we can't, maybe for some reason we don't have access to data about the system, and so we need to do some simulation to mimic what the data that the system would produce. And so with that, I would say always going back to working with whoever the domain scientist to make sure that your simulation makes sense. And so, like you said, inducing even various sources of noise is very well. I think in my experiences, I think what I used to do is that I would maybe generate 2 or 3 different kinds of data, one with maybe more noise, one with medium noise, one with maybe little noise. And then you model them, and then you see how the result that you get, whether they are similar or not. So that is one way. And then, but there's also sort of doing data simulation, and then there's doing data augmentation. So I think the augmentation part is tricky, because if you have some data and then you want to augment it, there are always ways of handling that. You can use Bayesian tools to let the model know that, okay, this part of data is augmented. And so maybe there are other sources of sort of variability that you should be sort of accounting for because they are augmented data. But I always advise that never augment maybe more than 20% of your data. Then you are doing more than you should be doing. And so augmentation is tricky. It depends on so many things. At times it depends on I ran into one example where, depending on your system. So for example, if your system goes this way, and you only have access to maybe one local peak, you can augment it in such a way that your resulting data wouldn't then reflect your population. And then your model will be driving you into a path that may not be the path that you want to go. So data augmentation is tricky. I would say that for me, for practice, I wouldn't advise maybe doing 20 or 30% data augmentation. And always there are Bayesian tools available. So if you can also use them to make sure you tell the model that, okay, this part of the data is also augmented. So the uncertainty quantification for those part of the data should be different. That is also helpful.
Great. Thank you so much for that answer. And thank you for everyone's questions and responses. Let's give the panel a round of applause. Thank you for being here today in person and our participants online. It's now time for everyone in the room to have a lunch break. And then for all our online participants, we will be sending out emails eventually with all of the recordings, but that might take a while, but stay tuned. Thank you so much.